clusters of square.
Great, welcome to the last day of this workshop. And throughout the week we have seen many approaches that drew inspiration from biology. So then I'm happy to invite Trym Lindell from the Oslo Metropolitan University, who will tell us about actual biological neural networks and how to measure complexity therein.
Yeah, as Bara said, we'll talk about a bit about part of how we measure complexity in biological networks. You might be familiar with some of the methods, but maybe it's interesting to see how they're sort of used in biological systems anyways. First off, like everyone else, also talk a bit about how I got to Alife. And it's been a bit of a sort of convoluted path because I actually started in psychology and fine arts. My bachelor's was like 50-50 psychology and fine arts. So I worked on something called neuroaesthetics, which is the sort of neuroscience and psychology of aesthetics feelings. But I also got this idea that I wanted to do modeling. I wanted to like make stuff already at the bachelor. My early intention was just like I need to get to like the point where I can like model the base constituents, the neurons. So I wanted to do network neuroscience or systems neuroscience. But the Mosers won the Nobel Prize, and I didn't have the grades to get in that one year. So I ended up with cognitive neuroscience, and then I actually found these like visualizations of deep neural networks features, like deep Trym, that sort of made me go into artificial AI. And I also had a brush with Game of Life because I was learning to program in MATLAB, and I was making like visualizations of the Game of Life in that. And since I wanted to get into artificial intelligence, I made poor leveling choices. So I had to do half a bachelor's worth of like assorted STEM, computer science, and maths. And also here I encountered one of the professors that was also working on cellular automata. So I got like another brush with that. And luckily I met Stefano, or I found his PhD announcement for a PhD in AI/neuroscience. And that's how I mainly got into like the artificial life ideas by making sort of taking the like let's make some synthetic living organisms basically, and also working in cellular automata. Now I've recently become an assistant professor, and now I have academic freedom to do whatever I want. I'm not sure what I will do, but probably be something with Alife in the future too. So just also this a lot of this work would be sort of as work or examples associated with the Living Technology Lab, with Stefano is the PI. So yeah, just to set the stage, the brain, real brain here. It looks kind of funky. Maybe the most complex structure that we know of. And this complexity comes from like many sources and levels. If you look at the brain areas, like this is an example of like the cortex can be parcellated into many different subregions based on different architecture of neurons, different neuron types, different like functional abilities and stuff like that. Each of the
So these regions are physical, and can you spot them in a dead brain?
Yeah.
Okay.
So basically, even from a very early on, you had the Brodmann map, which was based on staining slices of neurons. And there he basically would add a stain, he would look through a microscope, and he would see the different architectures of the different regions.
Sorry, question?
Yep.
Architectures, do you mean like connective maps or like physically different neurons?
Physically, both.
Okay.
There's like different number of layers in the cortex based on which area you're in.
Yep.
And there's specific neuron types in different ones, and somewhat different circuitry.
Okay.
Although there are like ideas of the canonical cortical circuit, which is like really within areas. You also have sort of specialization. So this is visualization of the orientation sensitivity of the striate cortex of a tree shrew, I believe. And here you can see that's like it's kind of like cool organized patterns based on what specific orientation of light these cells are sensitive to. At the bigger level, the different brain areas are connected through these like highways. So this is a method which is called diffusion tensor imaging, which let us visualize the direction of like the major pathways in the brain. So those are the white matter tracts, basically. And here you can see the pathways going in from the different brain regions, and also like the interesting corpus callosum, which connects the two halves of the brain. And this place, when you get even deeper down, you get down to like this like messy jumble of wires, which we are unable to see.
I think if you, yeah.
Ah, there you go. So you have like these sort of models, and you can have fluorescent microscopy of the neurons, and it's just it looks, interestingly, this looks kind of messy, but when you get into the details of like the connectivity between the neurons, it's like kind of sparse and specific often. If you look at the specific neurons, you also have like this crazy complex morphology. So there's many, many different neurons in the brain. All of them have very, very different kind of morphologies. These dendritic trees, which is all like the wiring you see, they can also perform computations locally. So you can have like direction computation in the dendrites. And you also have connectivity schemes, for example, apical dendrites, which are the top dendrites of pyramidal neurons in the cortex, receive a lot of feedback connections, and feed forward is more basal, connect closer to the soma. So a lot of interesting complexity there. So there's evolution from humans, but if you go to the fruit fly, it's also quite complex. Recently, we've sort of mapped out the full connectome of the fruit fly, the Drosophila. So this is also like freely downloadable. Yep.
So just a question for this. So I assume you use many fruit flies, right, for this connectome, or?
Actually, I don't think you do.
So it's
I think
for one specific one.
I think because if I remember correctly, on the dataset it says like specifically like female.
I see. Okay.
So because like one of the methods you use here is that you like slice one brain into tiny, tiny slices, and then you scan this, and then you connect based on like the sort of circle or yeah.
Okay.
So I don't remember how many neurons. I'll have it in some other figures. So we have this, and we have previously we had C. elegans, which is like a little worm, 306 neurons or something like that.
How many neurons is the brain? Is the fly?
For the fruit fly, I have it in a figure. I think it's like 119,000 or something.
100,000.
Yeah, 100,000. I think it's 119, but it varies a little bit from the specific connectome. We also have some other forms of biological networks that are not in brains. So this is the kind of stuff that I work with. This is an in vitro network made from dissociated neurons. Basically what you do is you take, in our case, these mice, fetal mice or rats, and you extract the cortex. You add a enzyme that clips all the connections. So basically you melt the brain, and you put it back in a dish on top of these electrode arrays, and then you allow them to reconnect, because neurons want to be connected with each other. And they form these also complicated, complex networks, which you then can communicate with and read out signals from through these electrodes. Some statistics, depending on your atlas, 140 to 260 different areas, local subregions within these, 100 billion neurons, 100 trillion synapses. By comparison, Llama 4 Behemoth is like 2 trillion, so we're not very close. So, why do we want to detect complexity in biological neural networks? Of course, there's like the general scientific desire to understand the brain. There's also a lot of clinical reasons. So, for example, some measures can have like one level in a healthy brain, and then they can degenerate in, for example, Alzheimer's disease. Also, specifically, some of the complexity measures that are sort of like that we call complexity in neuroscience are associated with consciousness. So you can use it to detect whether people are conscious or not, also the level of, which can be very useful in certain cases. And for our purpose, we also want to try and like optimize computational power of the systems that we work with, which is why I have been looking into this, and one of my colleagues have been working on this. So as I mentioned, what we're doing is we're building these, trying to build these biocomputers using biological neural networks. And we have had like 3 projects running after each other, where it's just basically attempting to do this. The basic setup looks something like this. You have data in, you have an encoder that translates the signal, your data, to something that the neurons can compute on, because we need to sort of speak the neural language. And then we need to decode it and get some sensible output from it. This sort of works, but isn't great at the moment. We have some examples like this from the Cyborg project. We have a biological network that's controlling a virtual robot. Yeah.
Is this using the controller? Is this using spike timing or spike trains?
I don't recall exactly what they did in this method. For when I did the decoding for chaotic time series prediction, basically we read out a array of we basically like time bin, and then we get an array, and then we just use a linear classifier on that. Yeah. So there's multiple ways of doing it. As you see, it goes in circles, so it's not super interesting. There are more complicated setups that can actually like do more complicated behavior with this. Like there is like the dish brain that can play Pong. A lot of my neuroscience colleagues are very skeptical about that, the performance of that paper. But also like my chaotic time series setup, it works without performance to control under certain conditions, but not by much. So
with such a robot, in theory, you can do like an avoidance behavior.
yes, so there's been many examples of avoidance behavior. So actually from very early on, there was a cool experiment where they took, I think they extracted the entire brain of a worm, and then they connected light sensors to some specific regions. Very simple mapping, just sort of like input-output mapping through a brain region. And then they had it do like light attraction behavior. And then you can also do obstacle avoidance.
What I mean is that, in theory, with 2 synapses, you can control such a robot.
Yeah.
If you have 2 sensors, let's say. So what is the limitation when you use biological neurons? Why do you just don't connect these 2 synapses and done?
Yeah, that's the question. Like that's part of why it's like this is like early, still a bit of early kind of research, because the performance isn't quite there, at least in the in vitro setting. Theoretically, you could do more complicated things if you use a full living animal, but then you get ethical issues. So there is so of course we have this like from all robotics. You have people who are paralyzed, you implant electrodes in their brain, and you can decode motor signals of robotics control for like robotics arms, for example, through the brain. So Neuralink is sort of making that more practical, but it's not new in that sense. DARPA had a project where they used a human in the loop to detect threats. So they would just sit and then like have images flashed in front of them, and then they had an EEG cap, and they would then use the EEG system to be part of a sort of threat detection system, which increased the performance. That's the most extreme case. One reason why we're using in vitro network is that we kind of want to avoid the ethical issues of doing that. There's also one experiment that's using a cat that's anesthetized, for example. So just images through the eyes, decoding of the visual cortex, and you can do classification with that. But that's a bit dystopian, so you don't want to do that. So this brings us to the issue, right? We have these in vitro networks. We've heavily perturbed them. We've melted them, we put them in a dish, they don't have an immune system, they don't have a support system. The connectivity is not formed through a developmental process. It doesn't have senses continuously, only during experiments. So all these things are like fucking up the way they work, basically. And so we need to figure out a way of recapturing that in the dish, which we can sort of try to approach by looking at these complexity measures. At the same time, we don't want them to be conscious. So for the conscious-related measures, we don't want that, which turns out I think is going to be very difficult. But because then you're like making a mouse with no mouth that must scream. It's, again, not something that I personally want to do. So for some of the measures that we're using, of course, we're looking at criticality. is one of the things. I guess most of you guys are familiar with this, but just to repeat, we have find criticality around the critical point of a second-order phase transition, and here we think we'll see a lot of like computationally beneficial features. Of course, one of them is, for example, dynamic range. So part of our issue is that we need to try and encode information. We need to encode it as well as possible to have a wide dynamic range, to have a lot of sort of variable input we can code on or compute on. I've been exploring stimulation timing. So basically, you encode information in the timing between stimulation pulses, sort of mimicking spike timing, which you know the brain is using for learning, for example. If you sort of don't care so much about the current criticality levels, you can do this encoding. So this is just different probe delays, different interstimulation intervals, and this is like linearly decoding back what input you put in. But and this sort of works at like a high scale. If you're looking at the confusion between different inputs, it's fine if you're on like the like big range of differences. But when you get into smaller ranges, you start getting a bit more confusion between the input. So, and this is like your range here is not very great. So one idea then is to try and see if we can have these systems become more critical to increase the dynamic range. So the brain does, in some cases, self-organize into criticality, which we can sort of measure, among other things, by like parallel distributions of neuronal avalanches, which looks something like this. So basically what we do is we measure the spikes coming out of the electrodes, and we bin them, and we look for continuous sequences of bins with activity, which is what is an avalanche. So these should then have a parallel distribution both in time and in size. And we had early work in this did find parallel distributions in biological neurons. So we know it's possible. Of course, the parallel distributions is not enough. You can get parallel distributions from other sources too. So you also look at shape collapse. The shape of the avalanches should be collapsible to the same shape if you scale them, which is another hint. And then also we look at branching ratios. So that's basically that on average, if you have like one neuron active, it should activate one neuron. This makes sense, right? Because if you have less than that, activity dies out. If you have more, you get a full burst, sort of reminiscent of like epileptic activity, which is not very great for computation. One of the issues here is that our in vitro networks sometimes do reach a critical state, sometimes don't. So we need to also find methods of tuning them. So usually, like when you start out, you usually would do this by fixing the ratio of excitatory to inhibitory neurons. You usually mimic the cortex ratio. But still then it's not consistent. So one of my colleagues, she worked on adding GABA neurotransmitter, which is an inhibitory neurotransmitter, which can decrease the activity. And what they found was that it actually did lead to criticality distributions or parallel distributions. And this, in turn, does lead to increased dynamic range, also in these in vitro networks. So the top here, this is from an experiment where they used, the encoding was ampere, so like the current strength. And in the middle, you have the critical systems, which is you have a longer range, and in simulation sort of verify this, although that makes sense. Bringing it a bit to some maybe more Alife related stuff. This is also a paper where they showed that if you have critical phenotype, so like this is a neural Ising model, if the activity of the neurons is critical, it's much easier to evolve better performance. So it has an effect past just this dynamic range that we're looking at. Another thing that we look at is also just the complex networks. measures. So like coming from graph theory and network neuroscience, because it is a network, right? And it's in the brain. This is it's, as I mentioned, very structured and has a lot of interesting features. Also looking at what characterizes the complex networks that you find in the brain from this perspective. So this is like generally sort of two approaches. You can do like a structural brain network, which you base on the structural anatomical connections, for example, with diffusion tensor imaging. Or you can create functional networks based on like correlation of activity with EEG or fMRI, or spiking activity. It's quite a lot of different relevant measures. I'll highlight a few of them, but one of the things I want to sort of highlight a little bit is that some of them are associated with integration, which is basically we sort of associate with short path lengths, for example. So it means that activity can be transmitted efficiently between different areas of the brain. And segregation. So that's, for example, clustering of like high interconnectivity of certain regions means that some of the processing is likely segregated and processed locally before it's integrated. And then there's also just like general features, like again we have parallel distributions of edge degrees. So if you're not familiar, edge degrees is just the number of edges that you have from a node. And in systems like C. elegans, you get something similar to a power law. There's some variation here in the edge degrees. We also look at clustering coefficients, so just looking sort of locally at the number of actual connections between neighbors over the number of possible neighbors. And again, in C. elegans, you see that it has this may not seem very large, but basically you do a comparison to a degree matched random network, and usually these biological networks have much higher clustering coefficients. Yep.
What's a degree match random?
So basically, you take a network that is basically like the same network of C. elegans, and you have the same number of like degrees of the neurons, and then you just like randomly reconnect it.
Okay.
Yeah. So a lot of like the networks measures use like null models that is based on like random networks or lattice networks,
Okay,
so there's some matching between the.
So like the actual connections of C. elegans are considerably higher than just like randomly connecting its neurons.
Yeah, it has a high cluster
Yeah.
coefficient. We also look at connection density. This has like a biological interpretation too. So connections are expensive metabolically. It costs a lot of energy to send signals. It's very expensive to maintain them, and of course, costs just keeping a connection alive costs you energy. So you want to minimize that as much as possible. So this is what you see also here in the Drosophila, the fruit fly. So from this connectome we had 139 neurons, and the connection density is quite low for the fruit fly.
Is it lower than other animals, or you say?
I think generally it's quite low. I just computed that for Drosophila and then for C. elegans. I'm not 100% sure, but I would assume it's also quite low. It's just for those two we have the full connectome, so it's that's like a bit more certain. Yeah.
Sorry, I don't know if you already said, but when you calculate the number of possible connections, is this like in a radius or is this all to all?
All to all. Yeah, so short characteristic path length is also a measure of like efficient communication between areas. Basically, you use some like algorithm to find the shortest path between neurons, and then you compute the average path length, shortest path length.
Like those are also related to the small worldness, right? Or
Yeah, I'll get to that in a second.
okay.
So the short characteristic path length is something you do share with random networks, but you don't see it in like structured networks. This measure can also be used to look for centrality. So that's like the number of shortest path lengths that goes through a particular node. And here you can also divide between like provincial hubs, which is hubs that are like the shortest path lengths in a local cluster, and also like the connectors that connect different areas. And this is also something that you see, like these particular brain areas are, I don't recall exactly, but maybe like the thalamus, for example, like does a lot of connections between different areas. Modularity is also something we see in the brain. Many ways of detecting it. So that's just to have high interconnectivity in local clusters and then sparser connectivity between clusters, which you can detect using different algorithms, among them like hierarchical clustering. And then we get to small worldness, which is then a balance of short characteristic path lengths and high clustering coefficients. So it means that you have this potentially this balance of integration and segregation. And see in the biological networks we have this relationship, but you also see it across many other different networks in the world. There are also other measures that you can use to like get a specific sort of number that characterizes this, but there's also some critics of it. Yeah.
Just to have a feeling, so what does this metric measure, more or less, this small world?
Yeah, so a small world means that you have like a lot of clusters. You have segregated local computations,
Okay.
but information can travel between these clusters.
I see. Okay.
So if we look at these, this is from in vitro neural networks from some of my colleagues. So this is part of like one of the decisions you're making when you're making this is whether you have like high density seeding or low density seeding. So how many neurons you have per square centimeters. And this has an effect on these measures of complex networks. And this here we have like a threshold for small worldness, and we see that high density networks is better. So this gives an indication that that's generally what we should be using. So what work in simulations too show that if you have small worldness and scale freeness, you can expand the amount of parameters within where you find criticality. So it can make sort of the critical area of the parameter space much wider, which is of course very useful. Yeah.
Yeah, just a question of terminology because I don't really come from that world. When you say scale-free networks, it's like networks with preferential attachment as opposed to.
It's about the scale-free distribution of the edges.
Yeah, but the way you build them is with.
Oh, how you build it? I don't. There is definitely an algorithm to build it. I don't recall exactly how it works.
Okay.
Yeah. So this is of course useful for us. We want to try to have higher criticality because we want to have better dynamic range and better computational power, and to get that we might want to try to induce these features in the network's connectivity. Another thing is where there's like internal interrelations. This is from evolutionary simulation studies. So if you have connection costs, so you add this metabolic cost to the connectivity of the network you're trying to evolve, and you need to do this on like some sort of problem that has modularity to it, you get better performing networks and you get more modular networks. So this is I always find it interesting that you like add constraints and you get better performance. It's also like counterintuitive. There's also like clinical implications. Generally think that a healthy brain should have like a mixture of all these different features. And you also see this, I mentioned this for Alzheimer's disease. So here you have like the clustering coefficient in Alzheimer's disease, top right is decreased if you have Alzheimer's, and the small worldness is also decreased in Alzheimer's patients. I will also say though that sort of the clinical use of this has been criticized a bit because a lot of people for neuroscience have been very excited about these complex network measures, and they just throw that on everything, and then it lacks a little bit of like interpretability sometimes. But for example, for this, it's kind of makes sense. So you have communication breaking down. So then, these are complexity related, but in neuroscience and psychology, we do also have like ideas for defining complexity, just straight what is complexity. And there's a whole bunch of different measures. As we were talking about before, like even if we're studying the one thing, that doesn't mean we're going to agree on how to measure things. So there's sort of theoretical measures. Some of them are usable to some level empirically, but a lot of them are sort of computationally intractable to actually use. And then you have some more empirical measures which you can then practically use, which has been used in a lot of cases where you're looking at like wakeful state versus induced states where you are unconscious. And I'll highlight some of these. So first off, just to get for some practical measure, is perturbational complexity index. This is very cool. I've been part of an experiment that did this. So you basically, first off, you need to scan your brain with an MRI to know where things are. Then you take a TMS, transcranial magnetic stimulator, which is a device that shoots the magnetic current into your brain. And it's very nice. Like when you test this, you basically find the motor cortex and you find your finger, and then you depending on how thick your skull is, you shoot increasing intensity pulses until your finger moves.
Does it feel weird?
Yeah, it's quite weird. Like your finger moves on your own. If you shoot in the visual cortex, you get like scrambled areas of the visual field. I also did a direct stimulation where you added like AC currents and stuff to the brain, and like everything flashes and stuff. So don't tell the university we played around with this. It's a very cool device. You can use it to also like do like temporal lobotomations and stuff, temporary lobotomations. But basically, you shoot this current in. You have an EEG cap on. You measure the EEG response. You binarize that into a binary matrix, and then you use that to compute the PCI. So the PCI is based on impulsive complexity. So basically, you're looking for like repetition in a binary sequence, but they sort of generalize a little bit so that it works on matrices. And then you can look both in the spatial direction or in the temporal direction. And then you also do some normalization to have it between 0 and 1. If you apply this to patients, you see that people who are awake and aware score higher on PCI than if you're asleep, specifically non-REM sleep, so you're not consciously dreaming, or a bunch of different kind of anesthetics. This is particularly useful, right, because some patients have something called locked-in syndrome. So it means that they are completely unresponsive, but they are conscious. And with this method, you can check if that's the case or not. Yeah.
So how would you test it?
Well, you just, so how you verify it is interesting. So basically, the way you can verify with locked-in patients is that some come out of it. So you do this test, and then you check afterwards when they came out of it, were you conscious or not.
But I assume that that's rare enough that it probably would never occur in the data set, right?
Yeah, it's very hard to get the data sets. I think this method has been tested on it, and I think that in a lot of cases when you have patients like that, you try to do experiments on them.
But it's just that patients are more susceptible for this happen. I mean, you might imagine it's stochastic. You don't know a priori which patient, unless there's patients who have proclivity to be locked in somehow.
Yeah. I mean, like people. What do you mean, like people getting locked-in syndrome in the first place, or people waking up in addition?
Well, I mean during surgery, waking up during. I mean, maybe you're just.
No, locked-in is a different kind of thing. That's just you have a brain damage that makes you unable to. So it's like a somewhat more constant thing. With the anesthetics, it's easy. You just put them under and then you.
Sure. But what I mean is, I don't know of that many recorded cases in general where people with locked-in syndrome come out of it.
Sure. Yeah.
So as such, it's usually not that easy to tell.
Yeah. No, it is definitely difficult. So that's why it's mainly these anesthetics methods.
I think there's also some like alternative methods too. I remember in my old university, there was a researcher by the name of Adrian Owen, which I think he had some documentaries about him. Yeah, there were some methods that they had where they would talk to locked-in patients, like with some tricky fMRI stuff. So they would ask them questions, and they'd say like, okay, if the answer is yes or no, if it's yes, like imagine you're playing tennis. If it's no,
yeah, exactly.
Something else, and they could kind of cluster which part of the brain is activating, and they would realize that okay, actually like a third of our comatose patients are actually kind of conscious in there.
Yeah.
So it's not the only method. You can kind of verify with other methods, I think.
Yeah, exactly. That's another interesting thing because like even projected expectations can show up as specific patterns in the primary visual cortex, which is decodable with fMRI. So that's also.
I see.
The next thing I want to talk about is where we're getting into sort of like neuroscience neural complexity ideas. So when we're making the in vitro networks, we're looking at like balance between excitation and inhibition to get to criticality. For neural complexity, they're looking at this balancing of information integration and segregation. So this is like the first thing. A lot of this work comes from Tononi and colleagues. And the idea here is, it's kind of intuitive if you consider like the visual system. So in the visual system, you have two different pathways. One that is sort of associated to what things are, and one that is sort of associated to wear and movement and similar things. And these are segregated pathways, right? They arise from the primary visual cortex and they go in two different directions. And you can have, you can sort of identify these by fMRI, but you can also identify them by, for example, people who have strokes in these areas. You can have people who stop being able to recognize what things are, specifically, for example, faces. You have patients who, I know there was one patient who had a damage to the movement-sensitive areas, and then her world is still images. It's hard to cross a road, it's hard to pour a cup of tea, because everything is just like still images. But of course, in a healthy brain, this is all also integrated. So you're perceiving me moving around, you're perceiving the identity, all in sort of one thing. So we have this balance does exist in the brain. So they start off with sort of defining or making this sort of generalization for mutual information. So this is, you have the entropy formulation. You take some system, divide it into two partitions, and the mutual information between those two partitions is the entropy of one plus entropy of the other minus entropy of the whole. And then the sort of integration is then defined as the sum of entropies of the elements of the system minus the entropy of the system as a whole. And neural complexity has a few different sort of formulations, but this is one where you're using the integration. So basically you have computed the integration of the full system, and then you divide the system into subsets of a given size k, and you compute the average integration for that size, and you subtract that from the expected integration at that size level, assuming like a linear increase. You sum that. It can also be formulated as mutual information between partitions of the system. Now, quite nicely, this kind of works depending on the system you're looking at. So if you have random data, you have very little integration. You also have very little complexity. If you have an ordered data, you have high integration, low complexity. But if you have this more complex kind of data, you actually get higher complexity.
How do you choose this K and the size of the sub?
Sorry?
When you do this...
You do all of them.
Ah, okay.
Okay, so ideally you do all of them. Practically, this becomes computationally intractable quite fast. So there are methods of subsampling and then correcting for the subsampling. Integration itself is a bit of a iffy measure because to accurately compute it, you need to have like the correct entropies, and if in empirical data, depending on the size, it's very difficult to get the correct frequencies of each of the states, possible states. So usually, often you can get like higher integration than it's supposed to be. There are like MATLAB toolboxes to compute these with the sort of improved measure variance. Also, for this is from the original paper with some different sort of schematic networks connectivities, and you can see here that for like the B network, which also they didn't like look at this, but sort of is a bit small world kind of potentially. You have high complexity and lower for the other ones. So my colleagues also looked at this in relation to branching ratio, some measures of criticality in association with like high versus low density networks. Not a very strong association, but one thing you can see is sort of like if you want to have high criticality, high complexity, that only happens when you're closer to 1 in the branching ratios. So while it's not very strong, it seems like there's like maybe like a necessity for it. Now, this is also like a kind of, I would say, a bit of an interesting topic, integrated information theory. This is where sort of it goes to. This neural complexity is where it starts. Then it sort of evolves into integrated information theory. We're currently at version 4.0 of this. I don't understand it well enough to do a proper lecture on it. But we'll look a little bit on it to sort of get some intuitions. So this is a theory of consciousness, not just how to measure it, but also sort of what it is. And they take this interesting approach where they start out with some axioms of about consciousness. And this is, I think, it's from the 4.0 paper. And then there are some purpose postulates from that, and that's sort of where they base the theory of. Then there's a lot of complicated math. In version 4, there's a Python package called PyPhi, which you can use to play around with this. But I will sort of talk a bit about how integrated information theory 1.0 works, because it's a bit more manageable.
So how are these, what are these version numbers? I mean, they are
These are like mathematical developments.
improved.
Yeah. So so some parts of it is, for example, as you see with this version, one of the improvements that this integrated information theory 1.0 sort of does over neural complexity, it deals with causality, because the other one was sort of like statistical dependencies. But it's developed to be like a huge beast at this point. So say you have, one of the bases for this first version of integrated information theory is what you call effective information. So say you have some system, you select a subsystem of the subset of the system, divided in two partitions, A and B, and if you want to compute the effective information from A to B, you basically give maximal entropy to A, and then you compute the mutual information between the maximally entropy A and B. And the idea here is that you're then getting like the causal effects between the two, or causal information between the two. This is not symmetric, so if you want to compute from one direction or the other, you need to do the same procedure. And so effective information of a subset is just the sum of both directions. And you use this measure to identify what they call a minimal information by partition. So you have your subset, you divide it into all possible partitions, and then you compute the effective information between all those subsets, partitions. And to do some comparisons, you need to do some normalization based on like the minimum, maximum entropy. And the minimal information by partition is the bipartition of a system that has like the lowest effective information. And this value is what you call phi in the original. So it's like the sort of key value from integrated information theory. And it's sort of like, it's an O because it's like this whole integration, and then it's the I from information. And phi is then the phi of a system is or a subset is the effective information of the minimal information by partition of a given subset. And then in turn you use this to measure, to identify complexes. So these are subsets of the system that is capable of integrating information. So if your phi is 0, then you're dealing with like two different systems that are not really having causal effects with each other. So basically what you do is you take all subsets of the system. Again, we're dealing with a lot of computational intractability here. And you compute the phi for these, and then you do this ranking procedure. Basically, you remove the subsets that are part of another subset with higher phi. What's I think is kind of cool with this is that it lets you sort of identify these clusters of causal interactions. So here, for example, given this example system, you rank all the subsets given phi, and you get these ranked complexes that correspond to these different possible subsets. Different network architectures here have different kind of values. So if you have homogeneous connections, it's both actually interesting. Homogeneous and modular connections have kind of like relatively lower phi values. And if you optimize a network to maximize for phi, you get this sort of more heterogeneous connectivity. And here they're using like top-based matrices to vary from homogeneous to modular. And here you get maximal complexity as like low intermediate levels. So kind of in summary, there seems to be some kind of relations between these complex network features, criticality, and potentially complexity, which is an issue for me, who wants to make ethical biocomputers. Because if I need to have like scale-free small world networks to easier get criticality, but that might be associated with complexity, maybe I'm making a conscious mouse brain in the lab. Yep.
Do you take that statement seriously?
I don't buy integrated information theory. I don't. So I don't think that integrated information theory is maybe the best theory of consciousness.
Yeah.
We don't have a good definition of consciousness. We don't understand it properly. There's a lot of good points in integrated information theory. There's a lot of good points in these complex ideas for complexity. I think it is worth being careful if you have high levels of complexity in a system you're studying. But I wouldn't be very concerned that we're actually making a conscious system. Because when you look at brain damages that can cause certain types of loss of consciousness, it seems like there are like not necessarily one particular area, but there are areas that are more important. For example, I think one very interesting kind of disorder is something called blind sight. So basically that's if you have your primary visual cortex damaged. You're consciously blind of what's located in that area of the visual field. Like if someone asks you, you don't know. You don't know what's there. But if there's a cup, your hand can grab it. If there's someone smiling, you will empathically smile with them. So the processing of it is there, but it's not being conscious. Of course, if you go for integrated information theory, that's sort of part of the predictions because that's one of the places you get some integration. So, yeah.
Haven't they also done some experiments on people who've had the connections between the two lobes severed, and sort of the same effect happens where?
Yeah, but that's also interesting because that again, like that's a case of like integration. So if you go from like the complexes idea, basically if you could do the analysis of identifying complexes, it would give you two different complexes that are not connected. Because when you kind of destroy the corpus callosum, you can't integrate the information between the hemispheres. And you become two people in one body. One can talk, one can't. One controls half your body, the other controls the other half of the body. And then you gradually diverge because you're getting different sensory inputs. So again, there are like these things that are still somewhat in support of some of these ideas, but not entirely. I also mentioned that there are still other problems. This is not the only problems we have with the systems. One is stability. This is another study we did looking at like are these dynamic ranges stable over time? They're not in vitro neural networks. The representation of input is also not stable. So even if we manage to like improve the dynamic range and improve all this processing, you still have issues with these systems beyond these complexity-related issues. All solvable if I threw ethics out the window and used a human with electrodes in their brain, but I don't want to do that. So yeah, this is a bit of an overview of complexity measures and how they're relevant in biological neural networks. Thanks to all the people that's associations that sort of support this. Any other questions? Yeah.
I guess I need to ask, what do you think is consciousness, or how do you detect it?
Right. I think it's interesting to look at consciousness from the perspective of what may be its functions. So I think, for example, it seems to do some kind of filtering for what goes into biographic memory. autobiographic memory. So maybe it's a part of the filter function. It's interesting that like what we can process within consciousness is limited. So like your working memory is limited to surprisingly small amount of information. I also wonder a bit if it has a function in relation to being a general problem solver. So for example, a lot of the things that humans can do, you can sort of automate. Like for example, there was a mention of Csikszentmihalyi, the Hungarian guy who did flow theory. And one of the things you see in flow theory, you use track of time, you kind of forget what you were doing, etc. And that's like if challenge matches skills. So you're sort of just using automation for it. And then it seems you don't really need consciousness that much. But when you're working with something new, something novel, something you really need to think about, you kind of need to be consciously focusing on it. I think maybe that's where I'm leaning towards, but I don't have a very sort of strong opinion on it.
So is this to say sort of consciousness is the active process of rewiring and learning? Or like it has a function in the active process? Whereas when you're in the flow state, you already have sort of the mechanism there, it's all wired, the network is set, you just
it's finished, yeah. So it's a process, that's sort of general problem solving that you do before you're automating.
Yeah.
I think an interesting thing is, although, which is pointed out by Tononi, is that the different parts of your brain are based on integration information theory more or less predicted to be relevant for consciousness. For example, so the cortical thalamic system is where they think consciousness is like highly likely to happen. By comparison, the cerebellum, so the little extra brain you have on the back of your head, it's mainly feed forward and not very interconnected. And I know of at least one patient who don't have a cerebellum. And they're completely fine, completely conscious. Interestingly, they need to consciously, actively do everything. So they they don't have automated walking. They have to think about every single movement all the time.
The heart beats, no, they breathe.
Those are like more brainstem associated things. And the heart has like internal circuitry for that. So that's, yeah.
Just on that note, the whole feed forward part of it is like, that's one thing the theory would predict would be a not conscious system.
Exactly.
Right? So like, if you're concerned about like a brain on a chip.
Use cerebellum neurons. Yeah.
Yeah.
That's
or like have like a creative way to like you have some like small modules that are at some point like connected to each other and feed forward like stacks.
Yeah.
So you just kind of again, if you really care about not making a computer that's conscious that can't do anything.
You have to be careful about not having any feedback loop. If you act to the environment and the environment responds to you, then you can say.
No, but I mean, so you and I have that relationship in that sense. Like I'm reacting to you and back, but you wouldn't say that you and I as one thing is conscious, right? What? With that theory, yeah.
If you say that, then you also say that language models are potentially conscious because they have a feedback loop. Yeah. Yeah.
This is sort of almost related to Alife. So you were looking at human brains here, but there's a lot of animals that have been shown to be conscious.
One argues that they have been shown.
Okay.
It's not.
They might be conscious.
Yeah, that might.
I think octopus is one of them.
If you do if you think about like the mirror recognition.
Yeah,
tests.
mirror recognition tests.
So that's for
they also engage in play and all of these things that might be a sign of consciousness.
Yeah.
But an octopus's brain is highly distributed. It's it's not in that squishy top bit. Really, it's within its tentacles.
Sure. Yeah.
So could there be like a distributed form of consciousness? Because here we're talking about like structures in the brain. But,
sure. Yeah. I mean, like if you can have like local complexes with high five, for example. That's entirely possible. I don't see any limits on that. I'm not a panpsychist, so I'm not on the entire like everything is conscious. But that's is it interesting. I like the integrated information theory people get confronted with the fact that you can make a graph with arbitrary high phi, and then you're like, so is this conscious? And they're just like, yeah, maybe it is. Maybe that's just the consequence of the theory, and we have to deal with that. An important thing is also that there's from the ethics sides, I think there's you kind of need to differentiate consciousness and the content of consciousness that is suffering and pleasure. So potentially you could be conscious without suffering. It's not because like there are brain areas there are areas in the brain that are associated with specifically like pain and pleasure and liking and wanting and etc. And you can actually go in and you can specifically turn those off. So there's this work by Berridge on dissociating liking and wanting. So basically, you can take a mouse, you can inject sort of local anesthesia kind of agent that reduces the activity in the wanting center of the brain, which means that if you have a mouse, you put its favorite food in front of it, it's just going to sit there because it doesn't want anything. But if you stick it in their mouth, they will have like the licking response, which shows that it likes it.
It's the Buddha mouse.
Yeah?
It's the Buddha mouse.
Yeah. And then reversely also you can have like, this is part of like the idea of drugs that removes your liking, like you don't have a liking response necessarily, but you have like intense wanting. So it's like dissociating these things. So if you just had a system that is conscious but don't have any of that, maybe that's admissible from an ethical perspective. Yeah.
Would you have any comments on the fact that, or on Roger Penrose's argument for the fact that consciousness is not computable? And he argues, yeah, okay.
Yeah, I know. I don't see any reason why we couldn't compute it. I also.
So he argues. Okay, so maybe I should mention one of the arguments, and one of his arguments is linked to Gödel's incompleteness theorem, and
I can address this. So it's a completely ridiculous. I mean, it's a mathematically good argument. Here's one way you can see this. Suppose that you imagine that everything that you care about in terms of a human or us can sit inside a very large box. Within a box, you can approximate what goes on inside the box with a finite number of degrees of freedom. As soon as you have a finite number of degrees of freedom, there's no girdle. There's just a giant, finite state machine. There's no problem. Like the girdle thing really requires an infinite tape. It really requires notions of infinity for it to make sense. There's just no way that has anything to do with consciousness. I mean, no way that I see it.
Yeah. Actually, better with the Chinese Roman experiment.
Yeah, exactly.
Yeah.
Can I ask a question? So I was just wondering, when you talk about these avalanches measures, I was wondering what is the metric that you're actually computing when you plot these graphs? So is it correlation or it's
no? It's so you get your spike time style and you time bin them, and then you look for sequences of time bins with spikes in them, and that's the length of an avalanche, the temporal length of the avalanche.
But so it depends on your binning, right?
Yes.
Or
Well, yeah, I think the idea is that if it's critical, the binning shouldn't matter so much.
I see.
You successively
if
increase
it's
the
not
bins
critical,
and see
some
that the
binnings
power
can
also
give
goes
you
down.
a power.
Yes.
Yeah.
Which is part of the issue. So that's why we also use like the shape collapse and branching ratios.
I see.
So you have a bunch of metrics, and somehow, if I want to generate a random graph that matches, n of the metrics, is it always the case that generically it will not satisfy the n plus first? I mean,
it depends on that. So like you can get criticality in a random graph, given the correct.
sure, you can do that. Yeah.
But a random graph would have short characteristic path length.
Yeah. So now, if you enforce that, it's like every time you can probably enforce anything when you.
It's like when you randomly connect.
But then you don't get you still there's a lot of properties that don't follow naturally from each other.
Right. Wait, sorry. Can you?
I mean, in the sense, let's say I want to create a fake brain graph. I mean,
Yeah.
Right. So I look at 3 of the criteria you mentioned, and so I kind of make it fake look real according to these criteria. But then generically, if I take the 4th criteria that I did not take into account, it will fail at that. If I did not explicitly, it's a bit of
Questions about the correlation between all these criteria.
Yeah.
That are
If I try
computed
to satisfy one, and create some fake data that satisfies this one, does it you know, am I likely to satisfy one that you have not
Yeah,
specified?
I think like if I remember correctly, for some of them I think are somewhat related. Let me see if I can get to the correct one. Yeah, so here I guess is an example of the setting up. I think like because they were focusing on sort of scale-free networks. Yeah, and then they sort of got small worldness from some of the other measures they're optimizing.
Okay.
But I'm not super familiar on like the exact relations there. Yep.
Yeah, I was just wondering, as someone who is concerned with animal consciousness, do you see the future when maybe the in silico models can replace these kind of experiments done on mice?
Potentially, yeah, if you have enough computational power. One of the issues currently is if you want to model, a single neuron to the complexity level of the morphology, you need like hundreds of differential equations just for a single neuron. And then that's not taking into account the internal cellular mechanisms that are affected by the activity and then is translated back into changes in morphology or synaptic weights or count, etc. Not to mention then all the other cells and the physics of the environment around them. So the computational power needed to simulate something like a mouse brain is so very immense that it's I think in principle possible, sure, but difficult if you want to do that detail level. Potentially you can do, or you can do a lot of similar kind of, or like we have abstracted away a lot of the details. So that it is in principle possible, practically difficult depending on what level you want to deal with. So that's it's definitely like it's unfortunate that you use animals a lot. Luckily for in vitro networks, you can use stem cell derived neurons. So that's often human, and you would use that in clinical settings. For example, you can grow neurons from a human with, for example, Alzheimer or something like that, and compare that to healthy controls. And while, of course, you're getting heavily perturbed networks, they might be different in some meaningful way. Another issue with these in vitro systems is that they're very, very unstable. First time we made some networks, which was clear from stem cells, everything died from a lab infection. The second time, 2 out of 6 died. This last time, all 6 survived, but their density was different from the second batch, even if we did exactly the same thing. So there's a lot of inconsistency, which makes comparison between systems very, very difficult. Even if it's the genetically identical mice, exactly the same procedure, you get different outcomes. So one of my main frustrations with this field is that you need a ton of money to make a lot of networks to get some averaging of some consistency, but you need to be able to make things consistent at the single subject level, because, for example, one of the things we were working with was making a protocol of optimizing them for some computational task, but for that protocol to be generalizable between the networks, they need to be more consistent. So you need to find ways of making that happen. So there's a lot of challenges in this before it becomes practical. There are people that are working on that are hijacking, so they use insects, for example, which implant electrodes into them, which can let you control their movements. You can buy a system like that off the internet from Backyard Brains, solar-powered cockroach controller. There's actually it's an interesting thing of the ethics of animal research. So the limit, there's a cutoff of what you like. That depends on how much you can do with the animal. So if it doesn't compensate, which means that if you take like an insect, for example, and its leg is hurt, it doesn't compensate when it moves. And it's the same for like zebrafish larvae up to a certain age. They don't compensate to not be more hurt by some movement or something. And in those cases, you have more freedom for what you can do. Past that, it's much stricter. With the insect case, I'm actually not sure if I buy it because they have like central pattern generators of movement, so they might just not have a choice in how they move. Any more questions? No? Okay, thank you.